Oh wait, don't I have to press go live? Oh, it's good. It's good. But it's big. It's all it's like huge. I don't know if you can see me. Maybe. But yeah, I see it. It's there. Can we watch? Do we have a watch stream? Okay. So do I have to click go live too or no? Okay. All right. Welcome everybody to the second Terp Genie Unfiltered. This is a special microbiology session, and we are delightfully gathered here today to talk about some interesting and potentially dangerous pathogens lurking in our society. And we're joined here today by Kyle Boyer. And Matthew Gates. I'm gonna let them give a little intro to themselves, but they're very familiar faces to all of you, so you definitely have seen them and maybe have heard from them, maybe have read some of their publications. But we are very very eager to jump into our discussion here today. And obviously we're running a little late. Persian Standard Time, people, it's a normie thing here in California for us. So thanks for staying tuned and standing by. Kyle, why don't you introduce yourself to our audience and kick us off here?
Sure. For those who don't know me, Kyle Ware. I've been working in cannabis testing for almost 13 years now, and actually, yeah, right around now is about 13 years. And currently work in cannabis microbiology with Biorad Laboratories. But I've done a lot of different things within cannabis testing slash adjacent fields. I currently serve as chair as the American Chemicals of the American Chemical Society's Cannabis Chemistry Subdivision. And yeah, I'm excited to dive into this topic because it's something I've been sort of following for a while now. And I'll pass it over to Matt now.
Hey, Kyle.
Hey everyone, my name is Matthew Gates. I'm an integrated pest management specialist, and I'm excited here to talk with Kyle about this particular topic about Fusarium and all this sort of thing. But for those who don't know, I'm the founder of Sentinel Consulting. I work in IPM support and agriculture, and a lot of times in cannabis lately, but also other crops. And yeah, there's a lot of nuance in this topic that I'm excited to dig into.
Well, we're certainly happy to have your expertise, Matt. I've been following you for a while, as well as following you, Kyle. So I'm really, really excited to have your expertise on this discussion because it's kind of crazy. I don't know what's going on here and why this became such a potentially disastrous biological situation for America. But obviously, what sparked this was the reports of some foreign nationals being caught in America with some potentially dangerous pathogens, specifically Fusarium graminearum. And I don't even know why this is a problem until I started looking into it, and until I had my intelligent friends start to kind of disseminate all of their knowledge to me, which ignorance is bliss, people. Okay, the less you know, the better off you are, because once you know what's out there, you're a little skeptical about everything you come across. So, uh, look, I mean, the U.S. raises a critical question for the United States, right? It's like we are potentially threatened by these things. So, why Fusarium graminearum? Like why not something else? That's the first question I have. And is this an actual biological threat? Matt, what do you think about this?
I mean, there's probably information I'm not privy to, but when I looked at the reporting, there are a few aspects that I think are important to note. Like, so the first thing is that the two people I have one article here from AP, and they talk about Yunqing John and Zunyong Liu, and they had these like baggies that had Fusarium graminearum in them, um, like explicitly, not like innocuously. And allegedly, they eventually said that the reason why they had brought them in was to do research. So it seemed like it was like an intentional um, accession, if you want to call it that, of this sample. And it's common to do research on Fusarium graminearum. It's super ubiquitous in many environments. It causes Fusarium head blight in wheat. It costs $1 billion+ in agricultural damage. So obviously you want to be careful about that. And we have organizations that are meant to you're supposed to declare these sorts of things, right? You would also declare things like produce. You'd also declare things like dried fruits and vegetables, nuts from certain areas, because if you don't do that, then some of these pathogens can get around. And we have historical examples like a chestnut blight. As little as the story goes, as I've been told, 6 trees were necessary to cause like billions with a B of trees to become destroyed in North America from a sort of Chinese chestnut that came in. And it had adapted to this fungus, but there is no such thing in North America. So things like this do make a lot of people cautious. But it could have just been that they just didn't want to deal with the hassle. Was it a bioterrorist attack in play? I don't think I have the information to make that kind of call. I would totally believe that they just didn't want to declare it, or they thought that they couldn't get it through the processing or whatever for totally research related means. I don't know.
Yeah,
I mean, but isn't it so ubiquitous that they didn't have to bring it here because it probably exists here? I mean, wouldn't that be like a reason not to have to bring sand to the beach with regards to that? I mean, and the other question is why Fusarium graminearum specifically? Like out of all of these different molds and potentially lethal or hazardous, pathogens, why that one specifically?
Whose research, then it would probably be for researching that one and not a different one.
yeah.
One would think. But I mean, I don't know. I'm not them, right?
Okay.
Yeah, I mean,
Okay.
they're focusing on this one species specifically, right? This is as far as like devastation for crops, like this is definitely an important one to look at because it does produce a lot of other things like mycotoxins, which we're going to probably get into in a bit here. But,
Yeah.
but yeah, I think, there's channels and ways to do this the correct way. So it is kind of like raising a red flag, like, hey, like you guys trying to smuggle this in rather than going through the appropriate channels for which this is done. Because if you're doing research, there should be no reason why if there's legitimate research going on here, there's no reason why you couldn't do this the normal way. But for whatever reason, they decided to take it a different route. So, and again, and how much of this is the media spinning something up, right? To create, alarm versus, okay, like, how big of a deal is this? I mean, again, as Matt stated, I'm probably not privy to some information. Who knows? But again, like Fusarium's like it has its issues. Obviously, it causes problems for crops. Is it going to kill people? Like, is this like is you trying to smuggle in anthrax or something? Like, no, it's not quite the same thing. I would say in that regard, but it has its own biological concerns.
Got you. Okay, that makes sense. So it's not like directly hazardous to people, just more economically speaking, and also for like the preservation of food and for different things like that. Like that in that regards, that's probably if it were, we're talking hypothetical, obviously, because we none of us have the information. We don't actually know. We just are privy to the news reports and the potentiation for this because of our foreign policy measures in the United States. So that's why it's concerning because it's like on what levels would there be targets here if we reach some kind of international situation with China, right, or another foreign nation. But it looks like to me that this is specifically something that could target the food economy and food generation and the practices that farmers are going through to like keep their crops alive and keep healthy and efficacious food in the supply chain. So that being said, what are the loopholes in the international situation for kind of bringing this stuff in? It's not like we're actively looking for it, but when we find it, it's such a big deal that the FBI is involved that this is on the front page of the newspaper. So, I mean, it's nice to know that these things aren't going to kill you directly, but why would this be so big of an issue that like the FBI had to investigate?
Well, okay,
I
so
would,
I
s-
go
oh,
ahead.
sorry.
Go
I
ahead,
would
Matt.
just say, I mean, if you wanted to get Fusarium graminearum, you could get it elsewhere, right? And if you wanted to do something nefarious with it, like there's not advocacy for it, but there are you wouldn't have to risk this sort of situation, so it is kind of odd from that angle, it's
Is
kind of
it
odd in that.
is it because Fusarium specifically creates the like and produces the most mycotoxins? Would that be the reason why this would be a specific strain of, Fusarium that would be targeted?
It is one of the higher mycotoxin producing uh, species of Fusarium. So yeah, there's a handful of them that do produce a lot of mycotoxins. Like this one produces deoxynivalenol, also known as vomitoxin. It's also produced
Vomitoxin. That sounds
vomitoxin.
amazing.
Yeah, that's a real fun one, right? So I mean, and the FDA does regulate these things, right? So depending on what type of foods we're talking about and what it's destined for. So like for human consumption, I believe it thresholds around 1 ppm. And then if you're feeding like this is in like grain and things like that. Now, if you're feeding it to animals, it's a little bit higher. I think it's like 2 to 5 ppm, somewhere in that range, as far as thresholds go. But so lower than the ones we currently test for in cannabis, right? So the ones we currently test for in cannabis, we don't test for these at all in cannabis, mind you, right? So those are at 20 ppb, and they're the aflatoxins, the sum of the four aflatoxins, B1, B2, G1, and G2. So some of those can exceed 20 ppb, and then we also test for ochratoxin A in cannabis, same thing, 20 ppb threshold. Um, so so yeah, but we don't look for any of these things in cannabis at all, and it is something that we see in food commonly, or not commonly, but it is a contaminant of concern in food. Obviously, nobody wants moldy weed, nobody wants moldy wheat. Same, but different. But yeah, so it's like just an interesting thing that there's this overlap, and that's kind of like what I've been starting to look into more is that um, I guess like when we think about like cannabis products, right, and how they're made, um, I think that there's like some interesting kind of things to look at. Like, right, we test for those five, analytes that I just mentioned, but we hardly ever see those actually. But there's
Hmm.
obviously a ton of other types of genuses of molds and fungi that grow are not only necessarily endogenous, but also just like can be found as contaminants within the cannabis microbiome. So when I see things like mold to gold, kind of makes me wonder like what else could be in there that could be problematic. So I mean, I feel like we've all heard of this case of somebody, I mean, and it's not Delta 8, right? Somebody hitting a vape pen and be like, oh man, like my stomach does not feel good, or it just ends up yakking. There could be a number of reasons for that. But I started to think like maybe it is the mycotoxin side of thing. Maybe someone just has some like really nasty material that they processed and distilled, and that's what's causing people to get sick. Again, so it's like one of those things where like I've been kind of probing a little bit further to try to better understand, uh, are these things in cannabis or are they an issue?
Yeah, and yeah, exactly. Mold to gold is really actually mold to mold, and these mycotoxins. Some of these things are, the fumonisins are definitely heat resistant, right? So there's a huge percentage of a chance that those are ending up in even these really processed oils like distillate that go through an extensive heat process that is, in a lot of senses is kind of like a pasteurization process for some of this stuff, but it doesn't really work to get rid of those mycotoxins, which are just the poisons left behind by the mold. So your mold might die, but there's stuff left behind that's more dangerous. And I think that's kind of the part of what is really scary about this stuff. It's like, maybe not on a huge level, but on a small scale, this stuff is really interesting to study because of these relationships. But Matt, what do you know, when it comes to pathogens like Fusarium, Graminearum, and others, are we kind of like a little vulnerable in the agricultural sense in this country, like to other things? Are we bound to see other issues pop up and generate in the future because of that vulnerability? What do you think?
Well, I'm a really big fan of using this concept I call the Everswarm concept. It's like this idea that like you have all the pests that you could possibly interact with whatever crop you're dealing with. Um, they're always constantly adapting. Insects and mites, especially the microbes, microbial pathogens tend to, sort of reproduce much quicker, and so therefore have the potential for adaptations to selection pressures much more quickly. In that way, I feel like it's always going to be like a sword and shield or a spear and shield type situation where we get a little bit better on the resistance end or on the treatment end or some level of the management spectrum, and then those selection pressures then inform what survives in the ecosystem or in the agro ecosystem, and then there's this sort of patty cake sort of interaction afterwards. But as far as actually being sort of threatened, the US does so much trade in agriculture that, yeah, it's a huge critical sort of like point where things go in and then come out. And, like I'm doing a huge deep dive into the budworm moths that affect cannabis and many other crops too. And, relevant to here, some of these moth larvae, they have adapted to be able to feed better on like nuts or flowers or tissues that have been colonized by these fungi. And they're very resistant to the toxins, but the toxins or the fungus breaks down the plant's tissues. So you get this kind of like symbiosis there. And then the moth, flutters around and transfers the mold elsewhere. And like what do you do when these fungi, these moths, they can like cross the ocean? It's true. That's how they got here into like the Americas from like Africa. They move up and down the environment, up and down the two continents. They do really well in the equatorial zone where it's hot, so they get a lot of generations. There's a lot of pesticide applications and things like this, and then they spread out and then they go through trade. So what we have to do is actively look for these. We are actually actively looking for things like Fusarium graminearum and other sorts of pests, but people try to surreptitiously move things like citrus and produce and other things because they bought it from home, or they really want to bring it back. They've never, they can't get it where they're from. I understand, but those kinds of things can like cripple, the food stores of the U.S. in this case. And it also affects many other countries as well that rely on this. So to me, I always feel like people should be very cognizant of that. But I think we have a very good system in place. It's just that it's always happening. The hyperadaptation is always happening. So we have to basically be on the same footing.
Right. And that cyclization of like how we're treating and how we're preventing and mitigating these things is the key, right? Like we have to be proactive in changing our techniques so that like we don't get overran by these things, or they don't like systematically evolve into like something worse that we can't necessarily treat as easy, which is another scary thing. It's a constant battle out there. So definitely definitely interesting, but thank you. So to just summarize, we're not as vulnerable as one might think in this agricultural industry, and we're a little better equipped than I thought, or I would consider even questioning. So that's good. But the question is also like, what about treating these things, and what about the survival rate with the current techniques that we're using? So like treating soil, treating the mold, whatever. If this can survive, do these techniques become obsolete? And then what's the future of that look like? Is it like GMOs is the only solution that are not susceptible to these things? Like what does that look like?
Well, I typically like to advocate for a multi-domain approach. And I think that genetics, resistance factors are probably the most efficient thing if you consider it from the farmer's perspective. So you have to front load the work and the resource allocation to make it happen. So in this case, you're whether you want to call it back ending or front ending, but you're putting it on the onus on the breeding and the selection for markers and for traits and evaluations of all of that. You don't even necessarily need to do genetic modification. This is just looking at the traits that are there and seeing how we can maximize them and optimize them for different environments, different regional pressures, getting that sort of kill chain really succinct is going to take time. We're not there yet, I would say, but it's really building. It's really building, and so in that way, I think that that's a good thing. But there's also many other things. We just can't be one-dimensional about it. We have to, like preferentially, you wouldn't be exposed to the pests, right? Like in the survivability onion, the first layer is, don't be seen. Don't have the problem in the first place. But if you do, well, then have a plan for those things. And another thing that cannabis in particular doesn't get is infrastructural support. I always talk about it, so I'm going to mention it here. There's more and more of it in certain ways in certain places, but generally speaking, like, a lot of this info, secret sauce that like in my field, like people are trying to like sell solutions or whatever. They talk about certain things that they may or may not be affiliated with. And it's tough because you don't have like a more or less unbiased or like a more empirical source that's usually subsidized by the government or some other organization. So people are on their own when it comes to these really complicated issues.
Kyle, what do you think?
I mean, just in general, like as far as like warding off the pathogen and things like that, and like if like what can we do to kind of prevent it, right? As Matt kind of stated already, like, well, the way I put it, an ounce of prevention is worth a pound of cure, right? Just keep it out in the first place, then you won't have the problem. So how does it get in the first place? Well, it gets in the first place where you have like any type of wound. So it's Fusarium pathology is actually kind of interesting. So it's a soil-borne pathogen, so it kind of starts from the bottom and works its way up. And it's kind of like analogous to like you had like clogged arteries, right? Like it's like a fungal artery clog, where essentially now like your nutrients don't get to where they need to go effectively. So we talked about the example of head blight with graminearum, right? And that shows outwardly. Um, but what's interesting about Fusarium, because it starts from that kind of like inside out sort of thing, um, it again starting from the soil. So you have like a wound or something, it gets in there. Now it's up all up in your xylem, and then but it doesn't really show anything outwardly unless it's the head blight, right? It typically manifests as fusarium wilt or some sort of dudding. And like, right, there's
Yep.
aside from the graminearum, we got things like oxysporum, we got Solani, we got other types of fusarium that kind of show this change, right? And that wilting or that dudding, it's basically just like a sad little branch. It's like, okay, well, that branch is not getting the nutrients it needs, so it's gonna be a little sad and have that fusarium wilt. Um, and so again, bolster your soil with other things. Bolster your, sprays, whatever you got to do. Trichoderma would be a good example of kind of
Mhm.
like beneficials that could help outcompete some of the, harmful stuff, right? Those are kind of like a good line of defense. But really, the thing is, if don't bring plant aids into your room. If you don't have to bring plant aids into your room, it's usually a good first step of keeping it out. Once you have it, I think it becomes a lot more difficult to combat once you have something like this.
Yeah, and I mean beneficials are great, but how effective can they be against fighting something like a fast-moving pathogen like spores? And also on what scale? Like what's the effective level? Like at what scale does that just not become cost effective, more just like a not a good bandage, like a
Preventative.
loose cause bandage?
It's a preventative, right? Again, like well, it's once you have it and it's actually gotten to the point of out of control, then it's like, well, putting some trichoderma in there is not going to save you. You might as well probably just start fresh at that point. And Matt probably would know a lot better. I'm not as a non-cultivator. Well, that's not true. I have cultivation a little bit, but as a non-professional cultivator, and someone who has like some background but no like really expertise, I wouldn't say I'd be the right person to ask about that. But maybe Matt, what are your thoughts?
I think you described a lot of things really well with regards to like there's different species, they do different things. And I know that you and I, we talked a little bit before this all, and a good point to make here is that like a lot of times Fusarium, people, it's in the soil. Like if you soil sample, let's say you're outdoors and it's in the soil, like a lot of people's experiences, it's like there, but is it at like a critical threshold where it's gonna be a problem for the plant? Does the plant have other symbioses going on that's sort of like deflecting or impairing this relationship from becoming pathogenic? Um, I did provide an image about symbiosis, and how there are even Fusarium species out there that are, and strains, I should say, that have been found to be not very plant pathogenic, but are actually hugely pathogenic to insects, kind of like a Buffaria or a Metarhizium, entomopathogenic fungus. So like, there's no like clear cut, concise thing. That doesn't mean that you should be growing Fusarium everywhere or whatever. That's not the miracle cure. But it just shows how like there's all of this adaptation. Like in the world of ecology, parasites become mutualists, mutualists become parasites, sometimes over time or in different populations. It's crazy and it's nuanced. But with regards to like keeping it under control, I've read like, there's not a whole lot of cannabis IPM research, unfortunately, because it doesn't get funded and there's all these restrictions, of course. But of those that I'm familiar with, especially recently in hemp, like in Kentucky and Tennessee, they're trialing products that are allowed, those that are actually allowed, like federally, or from a state perspective, like in the U.S., and they're not doing very well against like Fusarium graminearum, and also they don't do well sometimes against like leaf spot fungi like Septoria, and bipolaris, which has become a huge problem. In cannabis, it's not the same kind of pathogen, but people are caught flat-footed. They don't have any resources, and then of the research that we have seen, the resources that are available are a lot of times not efficient. They're just not effective enough. Whether they're used proactively or reactively. And
Yeah.
like what do you do as a grower who's invested all of your time and effort into this, whatever scale you're at, in the face of this information, it's not just kind of, it is scary.
Yeah, you can
And for
know
me,
there's
I want to
no
give
back.
people a salubrious solution that's healthful and isn't going to hurt people. But sometimes I don't even have something that I can recommend for new things that come out of the ether. Um, and a true professional wouldn't be afraid to sort of make that point. But then there's also people out there who want to make money off of other people's misfortune and also the dearth of information that's out there. And that's also, I think, a factor that people should be aware of. So like Kyle adroitly mentions, please do research and do that due diligence and try to find experts in that field. A lot of these people, they want to share that information, or you can find somebody who at least has adjacent information to share, like about Fusarium, for example.
Yeah. Yeah, totally. And I mean, like most things in cannabis, you have to kind of take the heuristic approach to learning, right? It's a lot of trial and error. It's a lot of throwing the spaghetti as we hear it called so often. But it's scary that there's not a ton of solutions, and it's scary that the solutions that are out there that you have to pay for aren't effective, maybe as effective, but like I guess that's up for debate whether they're not, or a little bit or a lot of bit effective. It's of course there's going to be data points to support either argument, I'm sure, and that's the problem also, is like we need some definitive answers. We need some really clear paths to success with things like this, because it's complex and it's always changing. So yeah, I agree with you, Matt. Definitely, if you're having issues out there, reach out and to the professionals that are supporting you, that'll help you kind of work through the issues and give you all of the solutions available that meet most budgets. But it's definitely a problem. So we'll see. And on that, it seems like a big issue. And I mean, I don't want to like compare it to something negatively like asbestos, but it seems like this is like the asbestos of the cannabis industry. And it's like, how do you remediate over time? Like, how do you get rid of those things with there being such bleak results with the current solutions? I mean, is there no real clear ways? Like, are we always going to live with this problem and just have to keep on top of it and combating it? Or do you think that there is a way through selective breeding, through potentially some other processes? And I'm not like a proponent for that. I'm just wondering if that's the only real actual solution is some kind of like genetic modification, some CRISPR technology that can effectuate the future change of this and mitigate this or limit this problem, maybe in one country or maybe across the globe.
I mean, like the example I would give, right, is like PM, right? PM's always been the bane of a lot of people's existence, and it's another situation where it's like, well, once you have PM everywhere, it, might as well just, there's not really a great solution. There are, band-aids, but not really great solutions overall. But breeding for PM, right, there's a lot of different like MLO genes and things like that. There are a lot of resistance genes and things like that that you can target to try and kind of optimize and yeah, breed for that sort of resistance. As far as Fusarium goes, don't know what those are. Again, this is like I've kind of focused in on I'm focusing in more so on the toxin side of things more than anything lately on the work I've been doing. But yeah, I mean, it's like, again, what do you once you find a target, then you've got a lot of breeding work to do, and then how do you get that into like a line and kind of actually make that a stable thing? But yeah, it takes time, and I don't think we know what those targets are even. And again, we're talking about the Fusarium species that are problematic, like the Graminearum and Oxysporum and others. But like maybe there are some Fusariums that are also like beneficial, that are endophytic, that are normally there, right? That actually, like,
Yeah.
kind of to Matt's point, like maybe actually doing something in symbiosis with the plant. We don't want to get rid of that, right?
Mm.
So again, it's like being able to differentiate between those things is kind of also a challenge as well. What if you, yeah, okay, great. I've knocked something out that, or like targeted, found something that makes it so that Fusarium doesn't get a hold as easily. But now I've also knocked out something else that is actually beneficial to somewhere in plant health. It's not a straightforward topic, right?
No, and I mean, you make a good point about the toxins. I mean, the toxins, we're going to really dive in more, but it's like some of those could actually be looked at as beneficial not only for plants but for humans. Like, I mean, like when I think of all these new medicines that they're creating to like, help people like Ozempic, let's say. The fact that trichothecenes are appetite suppressants, I just think that there's some crazy wackadoodle like doctor out there or some researcher that's like, I can make this into some kind of like Ozempic style and anoretic kind of situation, right? It's like that to me, my mind goes there instantly, and
Some of
I
these,
don't know if
some of these are immunosuppressants too. It's like, hey, if you need an immune
yes,
suppressant drug, right? I mean, some of these mycotoxins do that, right? So
right,
cyclosporins, right?
right.
Things like that.
Right. I mean, that's always the interesting thing about this biological world is that from this biological world, so we take so many things. I mean, most vitamin C, it's like created from a black mold, right? And most of these different products are created through this process. And I'm not saying it's bad or good. Obviously, microbes are great. Like it's part of this living world, and we needed to live actively with nature, and also it helps us as humans. But we live in such a sterile world now that I feel like and as a parent, I know, and I'm like the parent that's like, yo, rub your hands, like get some dirt on your hands, rub it in your bloody wound. You're fine. Don't worry about it.
Play in the dirt.
But it's we live in such a sterile world that I feel like everyone's looking for that sterile solution. So, I don't know if that's gonna work, but it's interesting to hear it and to hear what kind of put like organic solutions there are to just kind of help to keep this at bay. But it seems like we're also leaving a lot untouched with regards to the markers that we're testing for in food. I know that I've I've read some things, Kyle, you shared some things with me on Fusarium kind of exacerbating some clinical issues and genetic issues potentially with some people, and like CHS and like those mycotoxins. Maybe it's mycotoxin induced, maybe it's not just like a mold species, but it's something that's there that's interacting with the person when they're consuming cannabis that creates such a like a big problem. So do you have some insight there into like is Fusarium specific to like that I guess that exacerbation in
So,
CHS patients?
so let's kind of, so like you had mentioned genetic predispositions, right? So I think,
Mm.
and this is a hypothesis that was proposed in a paper by Zamir Punja and Kimberly Gwynn and Maxwell Young, where they kind of looked at a lot of the different Fusarium mycotoxins, and they said, well, wait a minute, like exposure to these, a lot of these, the overlap in terms of symptomology is pretty uncanny. Appetite suppression, nausea, vomiting, right? Obviously, deoxynivalenol is called vomitoxin, right? So again, there's like definitely some parallels there, right? But I think from the genetics perspective and from just in general what we know about CHS, right? CHS is definitely associated with people who are using cannabis continually over time, typically at higher concentrations and doses, things like extracts, right? Also, in my mind, that's might be where you would see more of these, right? Concentrate. You like to
Yes.
sell like you concentrate something, you're going to concentrate
Yeah.
all the other things that's in there, and it's not necessarily going to be desirable. But at least we've got like five different genetic markers for CHS, at least that we kind of think of as far as association. It's that TRPV1, the vanilloid receptor, right? Which capsaicin responds to, which is why that helps alleviate, we think, right, some of the symptoms associated with CHS. There's a dopaminergic component there, DRD2, catechol and COMT, catechol-O-methyltransferase, which is kind of like a dopamine reuptake. One of the genes involved with that. There's
Interesting.
also a metabolic component, CYP2C9. The transformation from delta 9 to 11 hydroxy THC occurs via CYP2C9. Maybe there's something broken there or some sort of issue there. There's a blood transporter, ABCA1. Um, I believe it transports lipids across the membrane. So maybe cannabinoids are also getting trapped right inside of the cell
Yes.
as well.
Yes.
So there's a lot of different facets to CHS, but what we do know is that it seems to be very much associated with repeated cannabinoid use. And you see this also with synthetic cannabinoids too, right? That synthetic cannabinoids can cause CHS without using any cannabis. Don't think it's neem. FYI, spoiler alert, Matt. And I
Yeah.
work on stuff with that too. But yeah, it is really interesting. So I don't think we have, as far as the fusarium stuff goes with CHS, the overlap's there, but and that's there's some correlation, but I don't think it causes it. But nevertheless, I thought it was a really interesting kind of point to bring up is that maybe this is what's precipitating that CHS. Maybe you get exposure to some of these fusarium mycotoxins, and then suddenly it's like, all right, you've tipped over the threshold of intolerance, and you keep adding in this poison or this toxin that I can't handle, and maybe that's what tips people over the edge. So I don't know. We don't have any, there's not enough data to kind of like kind of prove that out yet. But I thought that was a really interesting link that they pointed out in that paper.
Yeah, definitely. And I was reading a lot about like ionophoric mycotoxins too, and how
Oh
they're
yeah.
yeah, they're definitely responsible for like, inhibiting that transfer from cell to cell, and that information that our body relies upon to like really get nutrients, get everything passed through your body. It's like your cells need to not be inhibited by these things, and that's exactly what's happening with these toxins. So
Well,
it's
to add to the
crazy,
ionophoric piece too, right? If you're thinking about like epileptic patients, I mean, know
right?
most of this in the context of
Electro.
certain types of penicillium toxins as
Mm.
well, like citrinin or paxilline. Those are both those will both perturb your ion channels. And if you're using, say, for example, CBD as an anti-epileptic, and you've got it contaminated, and penicillium, by the way, also probably one of the more common, especially citrinin or Penicillium citrinum, is
Uh-huh.
one of the most common fungi found within the cannabis microbiome. So again, like there's a lot that we don't know about mycotoxins in cannabis. I'm just going to put that out there. Again, the authors I referenced earlier, I feel like are really leading the way in terms of, here's what we have in the cannabis microbiome, here's their potential to produce toxins, but nobody's really done the work to actually look into this. So while you may be taking a great, tincture, full spectrum tincture that's amazing, or taking an RSO and you're like, yeah, it works great, but God, I got this batch and now suddenly I'm more worse than ever. You got to
Yep.
wonder what those other factors are. I mean, obviously you have
Yep.
to get repeatability from extract to extract, unless you have like, very similar material, which even then there's, there's lack of consistency between materials as well, even if it's the same cut, or was it grown, yada yada, all those types of factors, right? But then you got to wonder what kind of contaminants might be in there, especially if you're, again, to kind of bring it back with the mold to gold thing. I mean, like,
Yeah.
who knows what the material is that's being processed, especially when it comes to CBD products. I mean, dude, when we're talking about hemp, like, bucket, chuck it, extract it, call it good. And like, and there's not like compliance testing that we have with regulated cannabis, right? Hemp is just like, all right, cool, below 0.3%. If it is actually, if it's
I mean,
actually hemp,
that's debatable.
Yeah,
questionable at
below
best, right?
0.3% of what exactly? THC
Yeah,
or trimethazine?
yeah, right.
No, that's so funny. I mean, that is, yeah, that is a funny thing about how our world works with all that. We're definitely missing a whole lot of stuff. And I mean, even from the fresh frozen, I mean, like if you're talking about concentrates, let's talk about fresh frozen. So that is where I think a lot of this unfortunate kind of stuff can come from for the extracts. I mean, we're talking like, you with the proliferation of like some of this equipment, large scale, now you're talking about larger lots of fresh frozen being harvested from like depth grows, from outdoor grows. I mean, there's got to be a little bit of something growing in there that's not cannabis, especially depending on what microclimate you're in. And that stuff mostly is going to two pathways, right? Two extraction pathways, which is solventless or water, ice, or butane solvent, hydrocarbons, whatever, propane, butane, however that's being conducted. And so that is the whole point of that is to preserve all of the volatiles in the plant, and with that, you're preserving those potential microbes as well, right? Because they're just being frozen, and then they're using a cold solvent to grow them. So we know that they're not like there's absolutely no heat in that process, so there's no chance that that gets affected there. But the filtration of that and what inevitably makes it into your extract, you're vaporizing that. So with solventless, I mean, solventless is definitely sieved through a progressively staged sieving, mechanism, whether that be like bags or stainless steel mesh screens or however. But like that's it, and with hydrocarbons, I think there's a little more of an opportunity to like clean this up at the micron level where you can like send it through like a progressively staged filtration that's got some pore sizes that are like sub micron, sub single micron level or something. So I don't know, that could be you might say that maybe, I know this is going to definitely shake it up for a lot of people out there, especially my extract boys, my solventless boys out there. Maybe there's a chance that you can actually clean up hydrocarbon extracts to a more efficacious level than just the solventless, because of the micron, the level of qualitative filtration that you're actually going through. That's just an opinion, maybe unpopular, but maybe that can help. Maybe that can help to mitigate the amount of these things making it into the extracts. But I don't think it's anything except forcing it through a small aperture to like prevent the whatever size spore from getting through into the extract. From solution, it seems possible through like a membrane style technology, but I don't know. Am I totally off base on that?
Well, Kyle, aren't a lot of these toxins, correct me if I'm wrong, I'm not a chemist, but like they're high, like Dalton, kilodalton size, like they're high density compounds typically, right? Or am I wrong?
Well, so I mean, well, they're not small. They're not all that small. I mean, it depends on what we call small molecules. I mean, they actually kind of some of them are
Yeah.
complex actually. But But I mean, I think what it is really is like you got to look at what polarity of your solvent you're using, right? I think, and that's where I think it's going to end up is ultimately like distillate, right? Because you're using ethanol, these things are all going to be readily soluble and things like ethanol. Um,
Sure, they're polar.
yeah, yeah. But as far as like, so the hydrocarbon though, right? Again, but
Hydrocarbon has a small amount of polarity, a small amount of polarity, but
you could
it's
still
mostly
potentially
not.
pull these things in, and right, it depends on how bad the contamination is as well, right? And also like kind of to bring it back as far as like, the levels of these things that we do find. We do still have some of those figures that we could pull up. Um,
Oh yeah, absolutely. Adam, if you can hear me, let's pull up Kyle's image one. You want Kyle or image two?
I think it's image two that we got. It's the one with the table. Because like so again, like we're seeing levels of
Image
these
two.
things.
Sorry.
Yeah, we're seeing levels of these things. There's a recent publication I'm going to show you guys here in a minute. I guess we could start from the beginning with the kind of the proof of concept. We can go image one to image two, but
Adam, do you want me to share it on my end? Oh, okay. It's on. Okay, okay. I'm watching our live stream right now. I don't see it, but we could be okay, there it goes. All right.
are we image
Um
one first or image two?
doing image. Hold on, I haven't seen it.
At the table. Okay, so yeah, this is the most recent publication that just came out. And this is out of Arizona State University. So I mentioned Max Leong earlier. He's a researcher there. He also was the one who kind of like pioneered this this paper with Kimberly Quinn and Zamir Punja, kind of looking at all the different things in the microbiome. But he just did a survey of 118 different seized both black market and gray market cannabis samples. And what he found in that is that there were 16% of these, so about one in six, 16% of them had mycotoxin contamination. And the ones that we currently test for, right, those four aflatoxins and the ochratoxin, that was not really it. It was other mycotoxins associated with aspergillus that they found. That was only accounting for 2% out of that 16%. 14% of those were Fusarium mycotoxins. So with that, like that's kind of wild to think about. Like, so it says to me that we're looking at entirely the wrong thing. And again, now this is black market, gray market stuff. So obviously, legal cannabis we would hope has a bit of a higher standard. Obviously, we all pay for testing. We all hope that testing is keeping us a little bit safer and that that there's not going to be things like that in there. But it is really interesting to know that like yeah, these things are a lot more common than we think they are. And again, regulations, I don't want to throw rocks at regulators. I'm not throwing rocks at regulators at all. Also, the regulators didn't write the regulations necessarily. Obviously, it's a bunch of lawyers and people who I guess consulted with scientists who said that this is like probably the best way to go about it. But we chose those five. The data is showing that those five are not really that prevalent. So some of the stuff that I'm doing with Yasha is showing that, flower samples, like a fraction of a percent is the failure rate. Detections, yeah, sure, we see some detections, and it's even questionable with the data that we've been working with if those are even real or not. Because just how people report things in metric, some at different points, people are putting in zeros. Sometimes people are putting in an actual number, but then those patterns change. So it's like it's hard to even say if these quote unquote detections, not necessarily failures or even real detections, could just be noise entirely. Um, but yeah, we don't see those five ultimately. Again, fraction of a percent. If you look at flour, that's what it looks like. And then so I thought to myself, let's move to extract. Let's see what the extracts look like. It's like 1.5 times more. So not that again, but ultimately
Not crazy.
that's not that high at all. So
What extracts though? In what
again, well,
extract?
this is all different types of extracts, right?
But average was about one and a half.
Well, no, it's 1.5 times higher. So if we go from 0.001% to 0.0016 roughly.
Right.
So it's a low level of contamination, but then you see papers like Max's that just came out, right, with this table, and it's like, whoa, wait a minute. Why are we focusing there when this is here? Right? Um,
That is the question. It's why are we missing the mark on that? Like, I mean, is it just because this is the most harmful five that they could find as like carcinogens or like
well,
what's that?
I think yeah, that's actually a very good way to phrase it, right? I think we went for what's the most harmful and what do we know that affects, other crops, agricultural crops of interest. Like we're gonna go for the ones that are definitely gonna harm people, right? And so yeah, 20 ppb threshold, very low level things, right? That's what we're going for, is the things that we know that are gonna really cause some serious harm at very low levels. And that's, when you first create regulations, it makes sense. Take a stab at what's going to be like the most harmful to society. But I think and this is where I think the work is really important. And like what we're doing is like getting that data and taking a retroactive look now at like, okay, well, all right, been doing this experiment now with legal cannabis. Why is nobody looking at that data to figure out, are our policies working or are they not, right? And are they actually doing
Because
anything
it's
to keep us
politically
safe?
inconvenient.
Huh? Sorry?
Because it's inconvenient. No one wants to say that they have a
Someone's
problem,
got to do the,
and
yeah.
there's all
Someone
these other problems going on already. It's convoluted and messy, and no one wants to be the messenger to get shot. That's my hot take, I would say.
I mean, well, kudos to Yasha for, not only procuring the data, but actually helping me crunch the numbers
100%.
out, right? Because again, like it, and sorry, instrument standard companies or people who have already validated an LC triple quad method for these five mycotoxins are like, damn it, now I have to have more analytes on my method or something else completely different. And I'm not saying one way or the other. I'm not a regulator, I'm not a lawmaker. This is just what the data says and where we should probably be focusing our efforts, maybe more so, because this is what we actually see that's out there. And so, yeah, will you maybe have to revalidate a method? I don't know. Again, that's up to the regulators to decide if they think it's deemed enough of a risk. But one area that I also want to talk about with this, right, is think about the FDA regulations for this. So I had mentioned earlier, right, in food and feed, well, like, I think it's grain. It's anywhere, and for animals, it's like anywhere from like 2 to 5 ppm is the range, depending on what type of feed we're talking about. Humans, it's 1 ppm. The levels that he saw in that study were higher, definitely over one, so in some cases for some of these mycotoxins. And then this is, keep in mind, the FDA regulations are for the oral route of administration, right? So oral versus vaped or smoking is a very different thing, right? You go through the
Yes.
liver with, with the oral route of administration, it's not as bioavailable. Shoot, if you're vaping this stuff, and again, these are heat stable, gaseous exchange in the lungs, guys, going straight into the bloodstream. And at those levels, right? What could it cause? What does it cause? Again, these are questions we don't really have good answers to. I can tell you right now, based on what we know, probably not great. And I have had plenty of examples of people like, yeah, I took a hit of my vape pen, and again, who knows where they got it from? Some sketchy brand, whatever, black market trap brand. They're like, dude, I was yakking, and I was like, I've heard this with Delta 8 for sure, but I mean, again, this happens too with just black market cards, trap cards too. So it's like, who knows? These are mycotoxins
Yep.
that mold to gold.
That is ultimately the question. One quick question from someone in our audience on our live. They asked what the sample set size was for the table that we showed. Do you know? Can
118.
you
There's 118.
180
Yeah,
Okay,
118 these samples. So they in the study they worked with law enforcement. Uh, they procured the samples from law enforcement. They had already, dealers that had gotten jacked. uh, and they ran some tests on it. And yeah, so again, like 14% of those were fusarium derived mycotoxins, 2% of them were non-fusarium derived mycotoxins. They're absolutely
crazy.
associated.
So nuts.
Yeah. It's a significant portion, right? So you gotta
I
wonder.
mean, 20, almost a fifth of the entire sample set, and most of those are Fusarium. So
Yep.
that's a little concerning, that's definitely one of the most or relevant and prevalent molds that we are encountering. But nonetheless, there's more stuff that's concerning that we have no, we have no clue on how that's affecting us. And the reality is, I think where kind of like the rubber meets the road for me on this is that like the immunocompromised and people that are using cannabis products, right? And one specific thing I wanted to actually address here, which is like, I don't even know if this is true. And I know that maybe we won't come to a conclusion here, but at least we can talk about it. But like Is it, so we talked about ingesting, we talked about vaporizing. What about local application? Like what about topicals? Is this stuff mobile through, because I mean, obviously we know that some aflatoxins, some mycotoxins are actually able to cross the blood-brain barrier. So like how does that affect people using topicals, using stuff like with the potentiation for having some contamination in there?
Well, at least I'm not a expert on this, but I did work in making transdermal patches at one point. So I know a little bit about this, right? You need the right level of skin permeation enhancers in there to actually get those things across the stratum corneum. That's that thicker outer
Yep, yep,
layer of your
yep.
skin, right? Um, I don't know. Frankly, I don't think that you're going to get these are if you look at the molecule, right, like they're they're not small. They're also kind of tricky. Um, if you're getting cannabinoids though through, and this tags along for the ride, very possible. Again, I would be most concerned about that vaped route of administration more than anything. Orally
Definitely.
ingesting, we already know is bad and when the levels are, what I had mentioned reference before, like, above 1 ppm, definitely bad. Again, definitely a lot larger than a 20 ppb threshold, but the fact that we're seeing those at these levels, and that's in flower, right?
Yeah.
That's flower.
And Kyle, was this all California flower?
California and Arizona.
California, Arizona. Great.
Yeah.
Wow.
Yeah, great. Yeah, I know. We're as all of us in California here on this call are like, great. Yeah, so that stuff might have a lot of fusarium in it. Awesome.
Well, but again, it's just the microclimate situation here just will breed these molds in a lot of cases. I'm in Santa Barbara, and Santa Barbara is like pretty much raining every day in October. So it's like it's not great because we have the fog, we have we're close to the ocean, so we have a marine layer that comes in. And for some of these farmers that are in the areas that are, pretty much always affected by not only the cloud cover but the moisture that comes with it. Certain strains and certain terpene profiles do really poorly in that environment, and then there's some that actually are pretty okay. And that is another kind of part of this discussion I wanted to try to get into is like there's gotta already be uh, some genetic predispositions of some of these plants that are actually going to hold up well to the environments and not actually, be like a host for these pathogens. Are you familiar with anything like that, Kyle or Matt? Like, have you been privy to any of these breeders or any of these chemovars that are like not susceptible to mold infestation?
I'm gonna let Matt take that one.
Well, I
Oh,
would
oh,
say that, yeah, I'll say that this that so like some pathogens I think are easier than others. But when we get these like super generalists, like the budworms I talked about, or like powdery mildews, various ones that we know in cannabis, or Fusarium, or botrytis is another good, or penicillium, like Kyle mentioned also. These are all extremely promiscuous fungi that are able to not only exist in many different regions and are able to infect many different kinds of tissues across the species we're talking about, um, but also they've got a veritable cocktail, an arsenal of plant manipulating compounds and hormone affecting factors, and they can disrupt or suppress the immune system. Powdery mildews, famously, I like to say, the ones that we know in cannabis, like something like 60% of their genome, so the collection of genes that they have access to, is for host manipulation. So they got really good at it. And fun fact, they come from a lineage of soil fungi that used to degrade plant tissue. So they went from like a what we might have called like a beneficial or a neutral soil fungus, and they, became more hungry for living tissue, and then started from there like 100 million years ago plus. So for these, I do think that genes is really important, but the problem is that the quicker or the sort of the when we make a change like this and we distribute to everyone, it's very hard for breeders to be able to sort of like deal with these confounding variables. Did you even verify that there is a white whale of mine is that breeders often, for good reason, don't really have access to the ways that you would verify this. And in traditional ag, if you go out and say that your plant is resistant to Fusarium or whatever, and then everyone who buys your plant doesn't have that effect, they tell everyone, and then you go out of business. You might even get sued for it. You might even be legally responsible for it in some cases. It sure depends. So there's like a huge onus on this. It's taken very seriously. People spend lots of money and time and effort to make things okay. And you can't be betraying these expectations. Meanwhile, where's the research? It doesn't really exist, or at least a lot of times in the US, or is heavily restricted. So I feel like we are you sort of behind the power curve in a lot of ways. And I want to be able to empower people to be able to actually sort of forge ahead. And there are some people who are doing this with like resistance breeding to powdery mildew. There are several resistance genes that have been identified. Then they have to be sort of bred with the kinds of plants people want to buy, right? So it doesn't, in some ways, it doesn't matter if the plant that the gene resistance is attached to isn't really desirable or doesn't produce enough. So there's like all of these other selection pressures that have nothing to do or very little to do, or sometimes are contrary to the things that we want in the plant production space and the cultivation. So like in those sort of situations, we really don't have, I think, the kind of arsenal that we have in other crops. But I do think that the technology being as advanced it is now, it might be quicker than ever to be able to find those resistances given enough space in order to do that. So that's the exciting silver lining that I have for people with regards to that.
I appreciate that. That was, I mean, definitely makes so much sense. I just, I've seen some strains that were naturally resistant to certain things like botrytis, for instance, as you mentioned. I
Like,
mean,
how did you find that out though?
well, we grow everything in the same area, and so there's an obvious, there's just an obvious visual, kind of test you do with your plants, and you're noticing that like I'm losing so many plants in the same tunnels as these other plants that I'm keeping, and I have a row of maybe split between two strains. And I mean, we all know that if your plants are right next to each other and one has botrytis, it's typically going to just go to the next plant, and it'll keep kind of infecting all these other clean and healthy plants as time goes on. Some of those strains just can withstand whatever pressure that's around them and not become susceptible to the pathogen, and it'll ruin the other crop next to it, just not that plant. These are all cannabis plants. It's not like we're talking about two different, types of like crops. It's all the same genus. It's all the same plants, just different chemical profiles that I think are responsible for, that defense. But that's just a theory. I don't know. Maybe I'm wrong. Maybe I'm right. Maybe it's partially that. Maybe it's partially something else. I can't think it would be like any kind of vigor issue. It's like these are all plants that are cut in thousands, by however many mothers that are being kept. So it's all coming from relatively clean plant stock, but not from seed or anything like that. So just a theory. And that's part of what this is. Obviously, it's not. There isn't actually any specific standard, which is quite ubiquitous in our industry, period, not just in the, biological sense. But like, dude, like where can we do some testing and figure this out? Maybe we need to get a hold of these foreign nationals, get their Fusarium graminearum, and we need to test it out ourselves. I don't know, but I feel like there's so much to explore with regards to these like, intricate nuances from plant to plant, and has to have some, maybe not to Fusarium, but like it has to have some defense or effect, whether it's botrytis or some other form. But I've definitely seen it, and maybe there's some other farmers out there that can attest to that. But it's interesting to me, and definitely there's no data to support that, but maybe we can figure some out together. So, I also know, Matt, you have a lot of information here that we want to actually share. So do you want to dive into some of your data that we have prepared? We have,
Um, with
your,
extreme respect and humbleness, this is somebody else's data that I have
oh,
Yeah.
I'm sorry.
taken,
I apologize.
brought
Thank you.
together
I
for
didn't mean,
people. You know, I don't want anyone to get the
a
wrong
true
impression.
professional. No, a true professional. Thank you for correcting
Gentleman
me. A
and a
true
scholar right
Yeah,
here.
professional.
no.
I
Yeah,
meant
including Kyle here, you know, gave me the kudos. He was like, you know, "Ah, this is such good research. I'm going to be mentioning this in the future, and people should check it out for sure."
that future is coming soon. For everyone that doesn't know, Kyle is he has a presentation at CanMed 2025. Kyle, can you tell us where that's being held?
Yeah,
Plug
so that's
yourself
it.
a little bit, my brother.
Wyndham Rio Grande in Puerto Rico, June 17th to the 20th. And so I've got a discount code if anyone wants it. CM Kyle B 25. So if you $300 off if you want to go. But for those of you
Let
who
me put
cannot make
that.
it, I mean, this is kind of like it's kind of why we wanted to do this, right? Because I know it's all the way in Puerto Rico. So for those of us in California and you don't have the resources, it's like conference ticket, flight, all that, right? Hotel, it costs a lot. But we wanted to have this discussion because a lot of what we're talking about today is going to be presented there. Obviously, there'll be more detail, more slides. I've got more tricks up my sleeve in terms of some of my own work that I've been doing, kind of investigating the subject a bit further. So thank you for that plug, Simon.
Yeah, of course, man. You gotta play it. But yeah, thank you, Kyle. And if you're near Puerto Rico or anywhere adjacent, please get there, check him out, and check out all the cool stuff that CanMed is going to be presenting on. But Matt, back to you. The info that you shared with me from somebody else that you want to go over and explain. Where should we start here? Because I you have a lot of cool visuals and data that I wanna present and have people see. You're
I
on
think
mute, Matt.
Kyle, yeah, thank you. I think Kyle teed us up really good for number one. We could just start with the number one from mine, Adam, which is the mycotoxin production potential. Um, Actually, let me just make sure that I can have a good look at this myself because the I
Right.
brought it up on my mobile.
Yeah,
Um,
this is exactly the paper that I was referring to, right?
yes.
Kimberly Gwynn, Zamir Punja, Max Leong. I mean, they're pretty much outlining it all out here for us as far as like, here are the different species of Fusarium. Here, and I believe I'm looking at the right one. Is that correct, Matt? We're just looking at just the Fusarium chart.
The
Which one
one
are
that's
we?
the
Yeah.
table five.
Table.
Beneficial Fusarium strain, is that the one?
No,
No, the one, number one.
Got you.
Yeah.
Got you.
yeah, so it is table
Adam,
five.
so okay, got you.
So yeah, you can see like all the different Fusarium species and the different toxins they produce, and you'll notice Graminearum's got a lot. Solani has only one. Sporotrichoides has got a decent amount. Oxysporum's got a decent amount, but it's all kind of like a spread, right?
Yeah, exactly. And you know, I like to bring this one up because you can see how there's different wait. Yeah, yeah, okay, cool. Sorry, I wasn't sure the mic was on, my bad. But yeah, you can see these are not all the species that exist, but these are the ones that I looked at. And you know, like for example, I had mentioned when we had met up that there was an example in the Soviet Union where a particular trichothecene, T2 toxin, was the result or the cause of thousands of people dying in the Soviet Union. And I did a little digging into it, and it looks like it was between 1942 and 1948. There were some famines going on before that time and a little bit after as well. Fusarium sporotrichoides is the Fusarium that caused this problem most prominently, apparently. And you can see here in the chart, it is here with the T2 toxin right there, right then and there. So they produce different things. Some of them produce a lot less than others, and I just think it's just a little good context graph. Number two, I'll just lightning rod these. lightning round, I should say. The picture number 2 shows some Fusarium.
Are you talking about the stem mycosis?
Yeah, sorry. I'll
Okay.
use the proper names that
One
I
sec, I
gave
just
in.
gotta put. We're on just a tiny delay, Matt, from when we need to put the thing up. So just say Adam, show image number 2 from the list, and then he'll put it up there. But Adam, if you could hear me, show image number 2, please.
Oh, excellent. Yeah, no problem. Um, yeah, so image number 2, stem mycosis, shows Fusarium actually colonizing the stem tissue of a cannabis plant and the branches, I guess you could say too. And like I said, we usually associate Fusarium as sort of a root pathogen, and certainly many species can be this and other things. But I think a lot of people have not encountered Fusarium in this light, so I think it's just helpful to give some examples where the pathogen is maybe sort of atypically colonizing. And a lot of times, and if I remember correctly, when this picture was taken and given to me, I believe they thought that they had wounded the plant somewhere here where you can't actually see it very well. It could be on the left here where this sort of white patchiness is with the gray. All of that gray are the spore producing parts of the fungus. And it's like, there's probably hundreds of thousands. I don't know if I dare say millions of spores in this like picture here depicted. But you get tons of these, and like you were saying, these spores often, they move to plants right next to them. However, sometimes things can be a little capricious, and plants used to produce spores before they evolved seeds. For those who didn't know, like ferns and that kind of stuff, and moss still does this. Spores have a lot of advantages. One of which being that you can make a lot of them for a lot less resources. The disadvantage being that you better have the right conditions. Otherwise, it's not going to go very well. So part of it is the grower. We have this thing called the pathology triangle, which has often been refurbished into the pathogen pyramid because there are other factors that we consider. But the big ones are the virulence of the pathogen, so the pathogen's capabilities, its physiology. Then there's the plant physiology. Of course, it has to be susceptible, not to pathogens in general, but it has to be a right match. They have to be a good host for whatever this thing needs. And then you have to have the environment, the cultivation procedure or the natural environment, however you want to look at it, and the human elements that kind of go along with that. And if all of these have the right puzzle piece edge, then you have the possibility for infection. If you take out one of these corners, then it cannot happen. So like to your point, we would want to do this kind of like multifaceted. You want genetic resistance to the pathogen that you're going to find in your region, and it needs to play nicely with all the other genes and hopefully be supported by other ones so it has a good defense. But maybe it's only good enough to suppress it, and maybe you get the pathogen. But then if the environment isn't very conducive, then that's okay, and it doesn't spread very well, on this plant and also to other plants. So even if you still get some of the pathogen, you would be able to manage it really effectively. And I think that's why it's important to say that it's IPM, integrated pest management, and not necessarily something more
IPS,
thorough.
integrated pest
Yeah,
solution.
IPS, integrated pest sterility or something. Yeah, exactly. So
No, I mean that was very well said. That was very well said. I mean, it's there's definitely more than one factor in getting these pathogens to infect your crop, but it's definitely good SOPs and hygiene is the key. Hygiene with everything. Clean, clean yourself, clean your plants, make sure that you have good practices. Call Matt, and I'm sure he has a nice SOP for you already written, and you can just save yourself the trouble. But it is definitely very prevalent, and it's everywhere.
Can I add one more
Do
thing here, Simon? With can we get a little
yeah, oh yeah,
conspiracy theorist on?
add away,
Can we
add
get a
let's do
away.
it.
little conspiracy theorist on them now that we're talking about at the beginning? We're talking about like, oh, these guys are smuggling this stuff in. So So here's a trippy fun fact. So, and I don't know if did we end up screenshotting this, or I guess Adam, are we able to pull up that New York Times? Do we get that over to him?
Which one was it?
Oh, I think I sent it to in our chat. And I don't
Is
know if it's too late to get it over to him, but I could just summarize it for everyone if they want. And
Yeah,
feel
it's a New
free
York
to,
Times article.
yeah, this is a New York Times article from 1999, by I think it's Rick
Wow.
Bragg, and it's called "A Fungus to Kill Marijuana Has Environmentalists Worried." Can you guess what happened here in 1999 when the Florida Office of Drug Control contracted a Montana company, a Montana biotechnology firm, to take Fusarium oxysporum, genetically engineer it to become extra virulent. Well, virulent's not really, or I guess infectious, I should say, and tailored it to basically eradicate cannabis crops. So they designed this Fusarium oxysporum, and these environmentalists are like, dude, law of unintended consequences. You're going to unleash this and then ruin everything else too in the process. So there's like the government has tried to weaponize this in the past before, to kind of aid in the drug war. There's a follow-up article about a year later, also in the New York Times, that talks about how they were going to do this to eradicate coca crops in South America as well. So again,
Wow.
like these are all just like, again, I'm not going to poke the bear with the government, but what I will say is that there's been some things that have happened in our past that make me go like, huh, is there a reason why all these things are around our cannabis crops and are infecting causing problems with our cultivation? Is it because we might have had some like we've seen things get out in the wild loose that we didn't want? I mean, hey, look no further than our pandemic, right? Um, Maybe, and something that I've always wondered about is like if we go and we sequence what actually does the oxysporum that does infect cannabis crops, can we trace it back to these Montana guys?
Wow. That's
Yeah,
very,
I would
very
say
interesting.
full-blown conspiracy theorist,
probably not.
guys. I'm sorry. Nah, Kyle's full of it. But I mean, hey, the New York Times article is there. Like, go read it. I've shared this
Yeah.
around with different people in the community, and they like.
I just put the link into our live chat. So if you're checking us out, check there. I will post this article on our stories later because it is fascinating. But that doesn't surprise. I mean, like, is it crazy that that doesn't surprise me? It's like
Not at all.
that sounds
No, it's not
exactly what
crazy.
yeah, it's like exactly what our government would spend money on. It's exactly why it was such an interesting topic in the first place when I read the article about the foreign national smuggling the Fusarium strain here, because ultimately it's like, yeah, like they know that this stuff has a giant effect somehow, economically, whatever it is. It is um, it is a saboteur kind of situation. It's
Hey,
like
go look
the
at the foreign
biological
nationals
stuff.
though. Don't look internally or domestically. Go look at the
Right,
foreign
right, right.
nationals,
Yeah,
right?
yeah. Don't look at our top secret studies and tests that we've done here, and the companies that we've paid with government tax dollars to produce these
Come on,
things,
dude,
because
follow
that's
along
what.
with the narrative. Follow along with the narrative. These foreign nationals are causing problems.
Oh yeah, no, I mean, and that's, the propaganda part of it is interesting because half of it is like uh the headlines are just there to like incite the fear and incite the blood boiling and bone chilling whatever effect of whatever they're trying to sell you today. But the reality is this shit is actually a problem. It's not that this is not a problem. It's that there is an issue with this stuff and it can really cause and wreak havoc if it's not properly like addressed and in communities that aren't tooled up or geared up to fight this kind of pathogen or problem and what that means. Like I don't think it really has a super effect on cannabis necessarily because they're just gonna keep rolling in the regs on us. It's not like they're getting rid of regulations anytime soon. But the fact that we're not looking really through the lens of, what's here and what's toxic versus just like, hey, what's the worst thing that could be here? And let's make sure that's not here. And I know that science isn't perfect and how we address these things isn't perfect. But I just, I feel like we should be doing better at societally, but that's just me.
But this is why we sift through the data later and take that retroactive look. Again, nobody has dang this, and again, like I said, I've already given kudos to Yasha for actually going out and getting it, and then not only that, doing the number crunching, but
Right.
like,
Kudos to Yasha,
why
definitely.
why are we not doing this in general? Like, look at all these analyses, and like, and again, this kind of brings back around to pesticides too, and like kind of the cat four movement and what we're seeing. You give people the 66 list of what you're looking for, they're gonna go find something else to use, right? And that something else may be way worse for us, and we're not even looking for it. So,
Good point.
guys, go, preference labs, go get that QTOF and do the full scan, and let's actually see what's out there
Yeah.
so that we know
Let's
what
get
we should be looking for. Because again, it's just like the with like analogs, right? Analog Act, right? Oh, they made this drug illegal. Let's just go ahead and make a one-off. Let's make a little chemical modification, and now it's a new thing, and law enforcement has to catch up and figure it out, right? So same thing here with pesticides. Oh, you gave them the list of 66. Now they know what we're looking for, so we're gonna go and do something else, right? So I don't know. It's a game of cat and mouse, but it's like, dude, like, why don't we learn from other sectors and other, what has history taught us? Why don't we start taking a proactive approach with what history has taught us, right? That's kind of the way I look at it.
Definitely. No, it's revealing this opportunistic kind of ability by these pathogens to change on us so frequently. It makes it hard to combat it without being kind of like without having such a scorched earth approach to killing everything living other than your plants or getting as close to it. And I know that all of that needs to change. I know that the symbiotic nature of cultivating food or cultivating some kind of herb, it's way deeper than the commercialization that we're all used to, and we need to kind of take it backwards. But who knows when or how that happens. But I feel like we're the first step is just having these really candid, revealing conversations with amazing people like yourself and Matt. You guys have been fantastic. I'm gonna leave it to you guys to kind of conclude here on this amazing um, the ability that we had to really talk through the details. I want you guys to just summarize, and I want you to kind of give it your final word, so that we can leave everyone with some really good knowledge and really something to look forward to.
Well, I'll go and summarize my conclusions. It's just like kind of conclusion slide of my CanMed talk, which is basically after surveying, and I looked at over 500,000 mycotoxin data points with the Yasha. So that's a lot of data points. Over 400,000 in flower, over 100,000 on extracts. We don't see very much ochratoxin A or aflatoxin, the sum of those four aflatoxins. We don't see it all that often at all. Again, it goes from a fraction of a percent up to maybe a fraction of a percent and a half, right? So not very prevalent to see these things fail. But what we do see is we do see a lot of these fusarium-derived mycotoxins, and there's a plethora of research coming out to support exactly what I've been saying for quite a while. Let's look at this more closely. Highly recommend looking into the work of Zamir Punja, Kimberly Gwynn, Max Leung. They're all doing phenomenal work in this area. Nicole Gauthier is another one. And ultimately, I think that we need to start surveying other things within the cannabis microbiome that are prevalent. Things like penicillium, right? Penicillium is the most common, especially citrinum. In general, penicillium is the most common genus of fungi found within the cannabis microbiome, which does produce a lot of mycotoxins. Nobody's really looked into this. So it's like, figure out, start looking at some of these things, understand if it's there or not, so that we can actually make policy that's making sense, that's actually keeping people safe, rather than just spinning our wheels and spending money and having them do the rigmarole for something that isn't actually keeping consumers safe or protecting anyone. So that's kind of my soapbox on that.
Amazing. Kyle, thank you so much. And big shout out to all of those amazing researchers that you were listing off there. Definitely check out all of their work and Kyle's work. Thank you so much, Kyle. Matt, what about you? Any nice wisdom, any nice morsels to leave our viewers with?
I think the key point that Kyle made, I want to echo, which is that we have to support these researchers and we have to be informed as a population of people who are consuming. essentially, and we have to hold some sort of like social obligation to whoever's in charge of making sure we do the right thing, legislatively, whatever. You have to be active and informed when you are trying to be active, and we can only do that with the people who are sort of pioneering this. So I really say shout out to those people. Um, Absolutely. And I'll just leave with this. It might have sounded a little bit doom and gloom from my perspective, but it's just the curse of knowledge of knowing how powerful these pathogens can be, or at evolutionary times we can see what's capable. But we have something that's very important that gives us the options that's possible, which is the mind. We're able to predict. That's the thing. So we take all these analyses that we've had, like Kyle has done and what other people have done before us, and then we can use that to inform our cultivation practice, to hold each other accountable in the community. I'm not trying to start any witch hunts or anything like this, but if you know that bringing in certain kinds of produce or whatever can cause problems to the environment, which obviously you should be invested in caring about, but also to the plants that you're growing for medicine, for personal use, or at a cultivation scale. These pathogens, and there's many good examples in cannabis, one in particular, these can get from a nursery to every cultivation place in the state and then into the nation and then internationally. And then they get into the home grower. Sometimes nobody is safe. And so we can only protect ourselves if we know what we're dealing with and we have research on what it is. And then from there, we can create the solutions that are possible. And then we all have to kind of adhere to it together, because if we don't, then we invite more chaos. So with that, I hope that people will be informed, try to become more knowledgeable, and then seek those solutions out with the people who are in charge of doing those things, whether researchers or legislators or whatever, wherever that's appropriate anyways. And I look forward to everyone's mutual success in that endeavor.
Thank you so much. And yes, we definitely echo that. We want everyone to be successful, and resources is the key. As you all are going about your daily lives, understand that below the surface at the microscopic level are some real threats for us. So as Matt said, it's all about the honor system of everyone adhering and conforming to these best practices. And time will go on, and we need more and more input from every one of you. It's super important that everyone get involved in the conversation. Everyone's voice, everyone's experience is relevant. There's no book or no standard yet. So add your knowledge, add your experience to the conversation. It can only help. Thank you guys so much. I don't want to keep everyone longer. There's going to be more information coming out on this, and we definitely will do a follow-up with our amazing beloved guests here, Matthew Gates and Kyle Boyer. We can't thank them enough. We can't thank our producer Adam enough either. Adam, thank you so much for sharing your skills with us today. But guys, keep your eyes and ears open. More Terp Genie Unfiltered information coming your way. And if you need to reach out to any of us, to our guests, Kyle, what's the best way to reach you? IG, email, what's the best way?
Email, IG, I mean, Canna Kaibo is my Instagram. You can
Canna_Kaibo.
Canna_Kaibo. You can reach out to me on LinkedIn. I'm on all the platforms. You just Google me, you'll know where to find me.
We just working on Discord, guys. We're gonna work on Discord with Kyle.
We're gonna work on Discord. Discord, you can use some work with Kyle.
But yeah, Canna_Kaibo. If you need anything, reach out. We can always get in touch with anybody for you as well. Reach out to us. And Matt, how can people reach you as well?
Aman, thank you for the opportunity to talk in this capacity. And if people are interested in learning more, you've been a great professional, and I really appreciate it. It should be said and should be appreciated. I didn't get that point out, and I'd like to. But you can find me at zenflamel.com for professional inquiries. You can reach out to me through the messaging system there. You can also find me on social media at SyncAngels, my personal account on Instagram, but more and more people are communicating through social media all the time, right? So you can find me there. I'm also on X and LinkedIn and places like that. So that's where you exist in the social media sphere, then you can contact me there.
All right, right on. Thank you guys so much again. Have a wonderful, blessed day. Terp Genie out.